Hey everybody, in this video we are going to take a look at Western blotting. So this is a technique that one would use if, let's say, you are a researcher and you need to detect a certain protein, a specific protein among a collection of proteins. And I kind of want to put this technique sort of within the context of, things we've already been talking about recently, let's say in like previous, I think just in one of the latest videos. So one of the things you might want to do before watching this video is review the video on immunoprecipitation. And that would be a lecture 12 video, and Western blotting, that's going to go in the lecture 13 video playlist. So now, again, putting this in context of something we've already been doing, or talking about, let's go back to this tagged version of a protein called CDC45. Now, this is something we talked about in the immunoprecipitation video, and I'll just review it briefly here. So here would be our promoter region. This would be the +1 site, so RNA polymerase II binds to the promoter region. This is going to be a transgene that we are going to put into a host cell like Saccharomyces cerevisiae. So the RNA polymerase II will bind to the promoter of the transgene, transcribe the messenger RNA, and this will be the +1 site. The +1 site is the very first base that is transcribed by RNA polymerase. This is the 5' untranslated region. This is the 3' untranslated region. This is going to be the start codon. This region right here, now if you remember, this is where we are going to have the codons for the flag epitope. You remember the flag epitope? That is specifically, what is the flag epitope? So it is a series of 8 amino acids. DYK, D, is that 1, 2, 3, 4, 5, 6? There's 4 Ds there. DYK, 1, 2, 3, 4, 5, 6, 7, 8. I think that's it. We don't need to memorize that. But essentially, it's a series of 8 amino acids that are recognized by the flag antibody. And we talked about this in the immunoprecipitation video, how we can tag proteins just by making a transgene like this with the codons for the flag epitope upstream of the codons for our gene of interest. So let's say, in this case, it's CDC45. So if we can get this transgene into a host cell like Saccharomyces cerevisiae, the messenger RNA will be made. The ribosomes will bind into the 5' untranslated region. We'll start translating at this ATG. We'll translate these codons, making the flag epitope. So I'm going to put flag here. And then there's no stop codon between the flag epitope codons and the CDC45 codons. The CDC45, remember, is a protein involved in DNA replication. So we end up with a fusion protein. So the ribosomes, there's no stop codon, so the ribosomes just translate all the way through, and there's the stop codon down here before the 3' untranslated region of that messenger RNA. And I'll make it a TAG. Now this is our fusion protein, and we talked about this because we were interested in, how do scientists study DNA replication and initiation of replication? So they have to purify all these proteins. And this is an easy way to do it, tagging the protein with an epitope, like the flag epitope. And then they can use certain procedures that focus on purifying the protein by focusing on this. Now, how can we use this to learn about Western blotting? So let's say, our goal is to get as much of this protein as possible, as much of this fusion protein as possible, because we want to use it in studies of DNA replication. Well, we have this promoter here, but maybe this promoter, is more active under certain environmental conditions. So we want to find the condition that causes the host cell that has the transgene to make as much of this messenger RNA and as much of this protein as possible. We're really interested in protein levels, so we're going to use Western blotting to look at the levels of the fusion protein, the flag CDC45 fusion protein. And again, the levels might change in different environments because it might be that the different levels may affect the promoter differently, or how often the promoter is activated differently. So we don't know. We're going to have to take a look at that. So here's a hypothesis or an experiment. So we want to do an experiment, and we're going to grow our host cells with the transgene in different conditions. Let's say we want to test 1% glucose, 5% glucose, and 10% glucose. We're going to try 3 different levels of glucose in the growth medium for the host cells carrying our transgene. And we're going to want to choose the one that leads to the greatest percentage of our target protein among all the proteins, right? So the goal is to determine which of these gives the highest percentage of flag CDC45, relative to all the other proteins. And we're going to want to grow. Once we determine which one gives us the most, well, that's the one we're going to want to use to generate large cultures to get as much of this flag CDC45 protein as possible. So we could use a Western blot, probably most easily to determine which of these conditions is best. So, how would we do that? Essentially, we would need to grow the cells in the different media. Then we would need to isolate total protein from, the cells from each batch of media. And then we would need to first analyze the protein by gel electrophoresis. That's the first step. Okay, so now gel electrophoresis of proteins is very similar to gel electrophoresis of DNA and RNA molecules. Usually we're using a different type of gel matrix. Usually it's an acrylamide gel instead of an agarose gel, but overall, conceptually, it's very similar. You apply an electric current. You have wells where you load the proteins into, wells that you load the proteins into. Usually you have a ladder. Just like a DNA ladder contains molecules of known lengths, a protein ladder is going to contain proteins of known molecular weights. So now I looked up one on the internet, just a standard protein ladder that we could buy from a biotechnology company. And the weights are in kilodaltons, and proteins are usually measured in the units of kilodaltons. So these are kilodaltons over here. The numbers aren't too important. 11, 17, 20, 25, 35, 48, 63, 75, 100, 135, 180. And so if we were to perform electrophoresis, we load our ladder into there. We separate the proteins through the gel using an electric current. The smaller proteins with of a lower molecular weight will migrate faster than the larger proteins, and we'll get a ladder that looks something like this. Now let's say we loaded equal levels of total protein that was isolated from our host cells carrying the transgene. And here, this is where we load the cells grown in 1% glucose, protein from the cells grown in 1% glucose. This is where we load protein from the cells grown in 5% glucose. And this is where we load protein from the cells grown in 10% glucose. And we could stain this gel with a dye that binds proteins. And what we would see is something like this. All the proteins of pretty much every imaginable molecular weight are going to be in here. And as long as we load equal amounts of protein, of the total protein in each well, we really shouldn't see any differences here. Because there are probably tens of thousands of different proteins in here, assuming about 6,000 genes in Saccharomyces cerevisiae, and some alternate splicing going on. It's going to be a lot of different proteins. So you can see right here that electrophoresis on its own isn't very useful for giving us information on which of these conditions produces more of the flag CDC45 gene. So, we could do a simple calculation on the sequence of the flag CDC45 and determine that it should be about 75 kilodaltons. So somewhere in here should be where the CDC45 flag fusion protein is located. So this is where Western blotting becomes very useful, because we need to actually, we don't care about all these proteins in here. We only need to know, how much of the flag CDC45 protein is found here, how much is found here, and how much is found here. So Western blotting can be used to do that. Essentially what we would do, and you probably see, if you read through the notes and you watched some of the videos, you saw probably precisely how to do this with a lot more detail than I'm going over here. But all of the proteins in this gel, this acrylamide gel matrix, now they're going to be transferred essentially to a sheet of what we call, called nitrocellulose. You could also use a nylon membrane, but traditionally for Western blotting, it's a sheet of nitrocellulose. Now, I like to describe this as essentially a sheet of paper that does not dissolve in water, right? So it's a solid membrane, but it's as thin as a sheet of paper. And you lie that over the gel, and then you run a current through it, and then the proteins move from the gel over to the nitrocellulose membrane. And there are microscopic pores in there, so liquid goes through, but the proteins stick to the membrane. Okay, so now you have all your proteins from the gel moved over to the nitrocellulose membrane, and now you're ready to detect the fusion protein. And how do we do that? So here's the membrane now, and now we can't see the proteins here. We could stain the protein with a dye, but that's not necessary to make sure it transferred. We could stain it with a dye, the membrane with a dye. We don't need to. But we can move on to detection, let's say to detecting the flag CDC45 protein. And how would we do that? Well, what we would do is we would incubate this with a primary antibody. That's the first step after transferring the proteins to the nitrocellulose membrane. Remember, there are 4 lanes here. This was the ladder lane. This was the 1% lane, 2% lane, 5% lane, and 10% lane right here. And all the proteins are here, right? We just can't see them yet. Okay, so we're going to incubate this membrane with a primary antibody. So we put a solution over the membrane and we shake it in the solution. The solution has the primary antibody. And what is the primary antibody? So the primary antibody, in this case, can be what we call, let's call it rabbit anti-flag antibody. And this is kind of, this is just a general structure of an antibody. We're going to cover these in a lot of detail in upcoming lectures. So this is called a rabbit anti-flag antibody. It's an antibody that binds the flag epitope. Now remember, the protein we're interested in has the flag epitope on it fused to CDC45. So our antibody is going to detect this. And what does this rabbit mean? So it means this anti-flag antibody was produced in a rabbit. So some biotechnology company injected a rabbit with the flag protein. The rabbit made antibodies to the flag epitope, and the biotechnology company took blood from the rabbit and purified the antibodies and sold it. So now we can use this in our Western blot if we buy it from the company. So essentially what will happen is that antibody will bind wherever the CDC45 is located. So I'm just going to put these X's there because we won't be able to tell where it's located until we do a couple more things. So the next step would be to rinse off that solution that was on there with the primary antibody and wash the membrane a couple times to remove any antibody that may have accidentally bound non-specifically to other locations. So we're left with the nitrocellulose membrane with all the proteins and the antibody bound to wherever the flag CDC45 is located. So now we have to put another solution on there. We can't see this yet, right? We can't see where the antibody is. We need to put another solution on there with the secondary antibody. And the secondary antibody might look something like this. So instead of just being an antibody, it's going to have linked to it, say, an enzyme called horseradish peroxidase. So it's an enzyme from the horseradish plant. I'm guessing it's an enzyme from the horseradish plant. Now, this antibody, what it does, let's say we can call this mouse anti-rabbit. Now, what this means is it's a mouse antibody that has been raised to recognize rabbit antibodies. So as a result, this antibody, when we incubate it with the nylon membrane in a solution, with a nitrocellulose membrane in a solution, and that nitrocellulose membrane already has the primary antibody bound to it, it will bind to the primary antibody, right? Because it recognizes rabbit antibodies. So I'm going to put another X here, here, and here. Okay, so we incubate the secondary antibody with the nitrocellulose membrane for, I don't know, 15 minutes or something. And okay, then we get rid of, then we dump off all the extra solution, and now we're on to the detection step. Okay, because we still can't see anything, right? So for the detection step, what we're going to do is we're going to add a chemiluminescent substrate for the HRP enzyme that's conjugated to the secondary antibody. So we're going to add the substrate in a solution, and the substrate will be found everywhere, but it's only going to be processed wherever the HRP enzyme is found. And the HRP enzyme is only going to be found where the secondary antibody is found. The secondary antibody is only going to be found where the primary antibody is found. And finally, the primary antibody is only going to be found where flag CDC45 is. So these spots right here will start glowing. So what we need to do is place this membrane under a very sensitive camera in the dark. And then what will happen, let me erase these spots, is these spots here will start glowing. Let me make this one a little bigger and these guys small. So everything else will be dark. Everything else here will be dark except for these spots right here. They will glow. Everything will be dark because every other spot, no other spots have that mouse anti-rabbit with the HRP enzyme bound to it. So the HRP enzyme is located here, and it's using the chemiluminescent substrates, processing that substrate and releasing photons as it processes it. So these spots are glowing, and the size of the amount, the intensity of the luminescence here is related to the amount of antibody that's bound here, and that's relative to the amount of flag CDC45 protein stuck at these spots. So we'll use our camera, our very sensitive camera, to record the light coming from the nitrocellulose membrane, and then we'll process the image. And what we'll do is we will invert the image. It's typical that we would invert the image. So every dark spot is turned white, and every white spot is turned dark. So our result would look something like this. So we wouldn't see the ladder here, but here's the 1% lane, 5% lane, and 10% lane. Now let's say we inverted the image, so we see a nice big spot here and smaller spots here. Okay, so we loaded the same amount of protein, total protein in each one, but we see a nice big signal for the 1% lane. We see a smaller signal for the 5% lane, and we see the smallest signal for the 10% lane. So if these were the results, you know, which condition would we say gives us the most, you know, the highest level of flag CDC45 protein, you know, as a percentage of the total proteins? It would be the 1%. Okay, so this would be this 1% medium, this medium with 1% glucose. This is the one we would want to use to make a really large culture of cells to collect the most of this protein to put through that immunoprecipitation process, so we could collect a lot of it to do our studies on replication. And you can see, hopefully now you can see why Western blotting is useful, because if you're just using a stain that can just stain all the proteins, you can't really see, you don't really know how much flag CDC45 is present in one condition relative to the other. But with Western blotting, you can clearly see that, oh, okay, there's more of the flag CDC45 in this condition relative to the other conditions. Okay, so that's a general overview of what Western blotting is used for, and you can use it for lots of other things too. Please, as always, let me know if you have any questions, and I'll see you maybe in the next video, or I'll talk to you in the next video. I think the next video we're going to take a look at the eukaryotic replosome. Okay, see you in the next video.